Hi, my name is Michelle Jaldin. I'm currently a junior here at Trinity, Washington University. I'm a biology major with a double minor in language and cultural studies and philosophy. After I graduate from Trinity, I plan to get my MD, my PhD.
My name is Sonia Garcia. I am a junior here at Trinity, Washington University. I'm a biology major and double minoring in language and cultural studies and biochemistry. After Trinity, I'm going to decide whether to go to med school or grad school, have more experience in research.
We helped launch a club here at Trinity University, which is called Ladies First, and it stands for Fierce in Research, Science, and Technology.
We are trying to help other women here at Trinity, Washington University have that drive that we also have for math and science, and we're trying to help them internships. We need more women in the math and science field.
And what better place to discover your strength at Trinity?
I'm Dr. Moitra. I'm a Clare Boothe Luce Assistant Professor of Molecular Biology. I get research funding from the Clare Boothe Luce and Henry Luce Foundation that has enabled us to set up our research lab at Trinity. We sequence the genome of patients with a certain rare disease called PXE. PXE stands for pseudoxanthoma elasticum, which we are sequencing the ABCC6 gene and certain modified genes. There's one particular phenotype, which is that there's calcification of the tissues. Tissues in the eye can calcify, and this leads to central blindness. So that's always a fear in parents of children with PXE. It's a very early onset disease. Kids as young as 5 or 6 years old have been diagnosed with the disease. So the earlier you get diagnosed and treated, and if you know what your mutations are, and if those mutations are associated with a particular phenotype, we can figure out how to modify your diet and make sure you get regular eye screenings if you have particular mutations. So that's why our work here is very important. We're actually using patient DNA. So after we've done the sequencing, there is a way in which we can tell them the results, so they'll know which mutations they have. We are helping real people here.
We are currently verifying mutations in the XYLT2 gene that's located in exon 4. To that, we are running PCRs and gel electrophoresis, and we are extracting our DNA from gel electrophoresis that we are later sending out to NIH so they can verify and give us graphs so we can actually validate our data.
We got interested in PXE research because my collaborator is Sharon Terry, CEO of Genetic Alliance, and she's also the director of PXE International. The reason why she got interested in the research was her children are affected with PXE, and she sends us DNA from PXE patients. We amplify the gene sequences and run the gels to validate that we have a product, and then we take it over to the NIH and they sequence it and they give us back chromatograms, and hopefully after we've analyzed the chromatograms we can detect the mutations in patients. But yes, we do have a molecular biology lab here. It's not perfect, and we have lots of little techniques through which we retrofit certain apparatus to do things like we take pictures of gels using our cell phone. We really don't have proper space in the setup to have an advanced gel imager, but once the new building is up, we'll have a separate research space with student benches. We can have lots of advanced equipment in there to basically run better gels and take better imaging pictures and maybe do other experiments as well. And we can have high-speed centrifuges, which we can't in the current building. In the new building, we'll have all the electricals up to date, but once the new building is up, we could probably do lots of other things, such as we can culture cells. We can't right now. We will have like cell culture incubators and laminar airflows, which keeps everything sterile. So currently we do the experiments on the bench top, but once we have the laminar airflows, we can actually do everything in a very sterile environment. So the new building would be great for research.
In this Biology 497 class, it has really made me the person I am currently because of all the research I have been conducting in class. Sequenced genes, I have extracted genes, I have ran gel electrophoresis, prepared buffer solutions in class. I'm also currently writing a research paper based on the data that we have concluded in class.
I got an internship at Children's working along with my biology advisor, Dr. Du Bois. We'll be working in the Center of Neurology Research.